Well, the chilly weather and the prospect of rain has driven me back indoors. Don't worry, I'll be back there out there again before the end of the semester. That's for sure. My subject today is a clearly uh, paraphyletic group, and that is the apterygote orders of hexapods. Um, Basically, the primitively wingless insects, those groups that evolved before the evolution of wings in the rest of insects. So you'll often see the term apterygote in referring to these orders. Sometimes in the older literature, you actually see that as a formal taxonomic name, Apterygota or Aterygota, depending on whether or not you want to pronounce that P in there. The name itself simply means wingless. A prefix from Greek meaning without or absent, and pteryx referring to wing. And we'll see that root pteryx in the names of lots of insect orders as we go along. You're probably already familiar with many, if not all of them. On my roster of apterygote orders that I want to talk about are the Protura, the Collembola, Diplura, and then two orders that go by a variety of different names. In the older literature, you'll see them combined into a single order, the Thysanora, but the prevailing opinion these days is that there are two distinct groups, the Microcoryphea, also known as the Archaeognatha, and then the Thysanora, which are also known as the Zygentoma. So let's walk through each of these orders and explain some of their defining characteristics and unique characteristics and how we think they're grouped together. So the Entonatha is an informal term to refer to the hexapods that have what are called entonathous mouthparts. Now, what I mean by that is that the mouthparts, the natha part of the name, are actually enclosed within an expansion of the head capsule. So what happens is that the sides of the head capsule grow forward, fuse underneath with the labium, and enclose the mandibles and the maxillae, and enclose them tightly so that basically the only mode of action is for them to be protruded through the opening, the functional mouth, and retracted. And then within the pouch of the head, they can be rotated and grind food between them. But that's what we mean. That's what we mean by entonathous mouthparts. This is contrasted with all of the other insects, the Microcoryphea, Thysanora, and all the pterygote or true winged insects that have ectonathous mouthparts. The mouthparts are exposed externally. Other characteristics of this group, and I'll have some images to illustrate these in a little more to help you to understand what the pictures I'm trying to paint with my words. You'll have some pictures for those. Another characteristic of them, an ancestral characteristic, is that they have what are called segmented antennae. In arthropods, the definition of a segment of an appendage depends upon the existence of intrinsic musculature within that segment. Intrinsic musculature are muscles that arise in the segment and extend out, and you see they arise and then insert on the next segment to have movement in that segment. So extrinsic musculature would be musculature that arises within the body and extends into the appendage. But intrinsic musculature is a muscle that arises within the segment and extends to the next segment. So in most insects, other than the ones we're going to talk about here, they have only three true segments in their antennae: the basal scape, the pedicel, and then the flagellum. In the entonatha, depending on how many total segments they can have more than these three segments, true segments in their antennae. Let's see, what else do they have in terms of plesiomorphic characters? They have abdominal styli, and these are probably the best guess of what they are is that these are the remnants of the ventral lateral appendages that are typical on all of the body segments of arthropods. And in in the species of this group, they have become reduced to form what look like short feeler-like extensions of the body. We also see abdominal styli in the microcoryphea and Thysanora, but they're also here in the Entonatha. And then the final characteristic I wanted to point out is that they have indirect sperm transfer. Perhaps you never think about this, but that means that these animals don't actually copulate. Rather, the male places either free sperm or a spermatophore, a package containing sperm, on either the substrate, or sometimes there's a glandular secretion that the male will place the spermatophore on, or in some cases, this mode of sperm transfer again is also found in the Archaeonatha, the Microcoryphea, and the Thysanora. There are some that produce a silken thread upon which droplets of sperm are laid. And so then the female, it's up to the female to take up the spermatophore or the sperm through her gonopore so as to fertilize the eggs that she's going to lay. So this is likely a plesiomorphic characteristic that has been retained and having direct sperm transfer and thus, and of course, internal fertilization. These guys also have internal fertilization. Involves copulation, where the male has specialized organs, modified appendages again, to place a spermatophore or to put the sperm into the usually the bursa, a sac on the female. So this is a picture of an SEM of a diploran head. What I'm trying to show you here are real pictures of Entonatha's mouthpart. So we have, let's re-grab that pointer here. We have here a dorsal view and then a ventral view of the head with the antennae here. This is the labium ventrally. And what you're seeing here are the expansions of the head capsule to grow forward and cover. You can't really see. You can see the maxillary palpi here and the tip of the maxillary galea here. But the mandibles and the maxillae are protected within this head capsule. Let's take a look at this from a lateral view now. So here we have that same diploran with the expansion of the head capsule down. It's going to reach down under and fuse with the labium. Then it extends forward. So here's the galea of the mandible coming out here, and here you can actually see the palpi on the side. You can't see any mandibles per se. The mandibles are hidden. The mandibles are inside of this pouch. And so this animal will extrude or protrude, because they're extruding forward. That's the protrude part. The mandibles and especially the maxillae through the opening here in order to scrape up food and take it into its mouth. This is an illustration. So this is from the same paper, and what they did was micro CT scans to rebuild three-dimensional models. Unfortunately, our library does not subscribe to this journal anymore, and so I could not access the supplementary material, because they would have more detail, perhaps even the 3D models we could have played with. Be that as it may, what we are seeing here are the three basal segments of the antennae in different views, and we can ignore this. This is the labrum, showing the musculature of the labrum. So what's these colored things here? The browns and that's sort of greenish, olive green there. These are muscles that are attaching to the segments of the antennae. So these are extrinsic muscles. These are arising within the head capsule and on the tentorium within the head capsule and inserting on this basal segment. Like this muscle originates back here on the head capsule and reaches up and inserts here. So when this muscle contracts, it pulls that point of articulation toward the head, causing the whole antenna to be raised. Now remember that the definition of segments in the antennae is that they have intrinsic musculature. And here's the intrinsic musculature I'm talking about. Here's one intrinsic muscle. Here's a second intrinsic muscle, the next most distal segment. And here's yet a third intrinsic muscle. And looking at it from the opposite side, now you can see there's intrinsic muscles on the inner side of the thing too. And so this is what defines the segments. This, by the way, this is also what defines segments in the legs and in the genitalia. So intrinsic versus extrinsic musculature. Extrinsic arising outside of the appendage in the body, and intrinsic musculature arising within the segment. So the first order that I want to briefly cover and to show you some images of is one that we didn't have. I don't believe we had any good specimens or any specimens at all of these in the teaching collection. And so the illustrations will, the pictures will be helpful for you to understand what these creatures look like. This is the Protura. I know that some people use the common name of coneheads for these things, but well, I have two complaints about that. One is that I'm not a great fan of common names because common names are limited in terms of their linguistic background. The common names we use would be English names, and there are other people in the world that have different common names. The other one is that I'm old enough to remember the Coneheads on Saturday Night Live, so demeaning these amazing tiny little creatures of the Protura with the name of coneheads just goes a little bit too far for me. So sorry about that. Protura, again, I'll talk a little bit and then I'll show you some images that should serve to better illustrate this. Protura, one of the unique characteristics about them is that they have no eyes, nor do they have any antennae. And as far as can be determined, all the evidence points that this is a plesiomorphic condition. That is, these things never in their evolutionary history had either eyes or antennae. Secondly, the first pair of thoracic legs function more or less like antennae. They're elongate and they're covered with sensory setae, and they kind of hold them up over their head and reach forward as if they were antennae. And so functionally, these animals turn out not to be hexapods, but tetrapods, because they're walking only by means of the mid and hind pairs of legs. Protura are also characterized by a growth pattern, I guess. Yeah, I guess that's a okay term to use, called anamorphosis, or one of my old colleagues back in the day used to call it anamorphosis. I don't know if there's a correct pronunciation for that, but my natural inclination is to call it anamorphosis. What happens here is that when a proturan hatches from the egg, it's got a head, it's got a 3-segmented thorax, but it does not yet have its full complement of abdominal segments. And when it's mature, it will have 12 abdominal segments, just to jump ahead a bullet point here. That's one more abdominal segment than any other hexapod has. And so again, looks like a plesiomorphic characteristic. The neat thing about anamorphic growth, is that with each immature molt, a segment is added to the abdomen. And let's see, the standard morphological description is that in a metameric animal, we have a series of segments in the body, and we have a non-metameric anterior part of the body called the acron. And when I say non-metameric, what I'm saying is that in embryonic development, it is not characterized by having a mesodermal sacs that define the metamere. So a non-metameric portion at the anterior end of the body called the acron, and a metameric piece of the body at the posterior end called the telson. So when at the molt, the proturan immature adds a segment, it's right in front of the telson, right in front of that posterior non-metameric piece of the body. Can't say segment because it's not a true metameric segment. So this is the only group in insects in which it occurs. Just as a side light, because I don't have this on the slides anywhere else, and I think it's probably useful to mention it. Once a protoran reaches maturity, that is, it's sexually mature and can reproduce, it no longer molts. This is also true, as I'm sure you're aware, of pterygote, that is winged insects. Once they reach the adult stage, they no longer molt. But the other apterygote orders, the Diplora, the Collembola, the Thysanora, the Microcoryphea, they do continue to molt as adults. And sometimes these things will molt dozens of times in the course of their lifetime. So this is an interesting difference in these very basal hexapods that you don't see in the winged insects. Let's see, I talked about the 12 metameric, 12 abdominal segments. Protura is not, it's not a large group in practically all senses of the word. They're extremely small animals. You're talking about a millimeter or less in length. There are very few species. Well, very few in the context of arthropods. There's only about 500 species or so. Although I will say that because of their small size and cryptic habits, they live down in the soil. It's quite conceivable that there are going to be lots of newly discovered species of Protura as we move along. They're worldwide in distribution. They're really almost everywhere in the world, but because they have these cryptic, they're small and they're cryptic down in the soil, most entomologists do not encounter them. Perhaps more likely for an acarologist to encounter them when they use extraction techniques to get mites and such out of the soil or the leaf litter. That's where you pick up proturans. So I've got some pictures of proturans, but I want to give credit to where a lot of these pictures that I'm going to show you have come from not only of Protura, but Diplora and Collembola, and the other two orders as well. It comes from a website called the Chaos of Delight, which just has some absolutely beautiful imagery of these groups of hexapods, things that most people only, well, they may never see them, or they see them only as alcohol preserved specimens in a course like this, in which case a lot of the beauty, I mean really the beauty of the animals gets lost. So let's take a look at some of these. So here we have a proturan, and let's see what we can point out here. First of all, here's the mid pair of legs, the hind pair of legs, and these are the forelegs that are being used not to walk on, but as a sensory structure. You can see it's got a long segmented abdomen and fairly weak sclerotization and melanization of the sclerites in the abdomen. Remember, at maturity, it'll have 12 abdominal segments in addition to that apical telson. Here's the head capsule here with bristles coming off the front. Those are not antennae. Those are just sensory hairs. Here we see, let's see, a couple of things I want to point out here. Again, this creature is now going from left to right, holding the forelegs over the body and tapping. You can see the long sensory hairs. Exactly what they're sensing, of course, I don't know that anyone knows. These are quite probably at least thigmotactic, so they are sensitive to touch. But whether or not there's any chemosensory functionality in these setae is unclear. One thing, look closely. So we see the middle legs, the hind leg, and this thing right back here is a protrusible organ that is found in the abdomen of many, not only in Protura, but in many of the other insects and hexapods we're going to be talking about today. So they have these eversible glands here on the abdomen. Here's another one. Again, the small size, forelegs reaching forward as sensory structures. There's not a whole lot to say about Protora. There's very little known about them. They're detritivores, maybe herbivores or fungivores, things like that, feeding on whatever's available in the soil. Now, in contrast to this minute, very poorly known group, let's go to one which I suspect is very familiar to all of you. Oh, sorry. Wanted to show you the front end of the head of a protoran. Forelegs with an elongate CD. In here, you see that the mouth parts are not visible. They have to be extruded, that's the labrum here, through these openings here in order for this creature to feed. Now let's move on to the collembola. Remember from the Misoff et al. Article that the transcriptome evidence from that study tells us that the closest living relatives of Protura are the collembolan, and that together they form a monophyletic group with all other hexapods as a separate monophyletic group. So collembola are absolutely rich in apomorphic characters that define the order as a monophyletic group. Instead of having a 12 or even 11 segmented abdomen, in the collembola the abdomen is reduced to a maximum of 6 segments, and often these segments can be fused together so that there's no visible segmentation whatsoever in the abdomen, sometimes even being fused with the segments of the thorax. There's some controversy over whether or not they have compound eyes. Certainly they have clusters of ommatidia with which they can see, but they don't have the very discrete, tightly packed ommatidia that is typical of compound eyes in insects. One of the really distinctive features about them, although I must admit that some collembola have lost this structure, but that is the spring. On the 3rd and 4th abdominal segments is a ventral organ, probably the modification of appendages on those segments of the abdomen that functions as basically a locomotory organ. I'm going to show you a picture rather than try to describe it in detail here, but the two functional parts of this are a fork-shaped furculum that arises on the underside of the 4th abdominal segment, and it folds forward and articulates with structure on the ventral side of the 3rd abdominal segment called the retinaculum. As you can infer these functions of these things, or you can infer some things about these structures from their names. The furculum, by its very name, is fork-shaped, and so this is commonly bifid, and when it's folded under the abdomen, articulates with the retinaculum, which holds it in place. I'm going to hold the description there for a picture. But let's move forward just a little bit. Another ventral abdominal structure that characterizes the collembola is a structure called the caulophore. The caulophore is a bit of a misnomer. The name literally refers to containing or holding glue. It was thought at one time that the structure was used to enable the animal to adhere to the substrate. In fact, it's probably a water balance organ or a homeostatic organ to keep the water or the salt balance relatively constant within the animal's body. This is located at the base of the abdomen, but again, on the ventral side, just like the furculum and the retinaculum that are forming the spring. There are tons of species of collembola, over 6,000 known, and that doesn't even begin to scratch the surface of the species level diversity of these things. And in terms of numbers of individuals, they are extraordinarily common, typically they're found almost everywhere, but they are very typical of the soil, litter, the substrate, being edaphic creatures, but they can be found everywhere living. There are some that live on the surface of water. One of the very famous species is called Podura aquatica. It's a tiny little thing that occurs in vast numbers on the surface of fresh waters. Some collembolans also occur near salt water, near marine environments. They don't get out into the free ocean, and they don't go under the surface. They live on the surface along the edges here. There are some collembola that live externally on plants and feed on plants, and even some of these that can be common enough to be pests. There's one that feeds on alfalfa. That's a particular pest. Well, I'm familiar with it from Australia, but probably in other parts of the world as well. So there's a lot of difference in their life history. Basically, these things are either herbivores, fungivores, detritivores. I don't know of any predatory collembolans. So let's take a look at some images of collembola and see what they look like. So this line drawing is intended to, I put it here in order to show you where the furcula is and where the caulophore is. So here's the caulophore, COL, pointing to this extrusible element, probably homologous with the same thing that I showed you in the Protura, that functions in water balance in the collembolan. Let's see, hind legs, middle legs, forelegs. These are the segmented antennae. And in this group, this one has a clearly segmented abdomen. It's got, let's see how many, 1, 2, 3, 4, 5 visible segments. There might be a small sixth tucked in underneath there, or perhaps it doesn't have a sixth. This is the furcula arising from the fourth segment. Actually has a very broad base, but basically it's coming from the fourth segment. It folds up under the body and is held in place by the retinaculum. Now, I talked about Sydney Manton last time. She studied the locomotory mechanisms and the musculature of collembola to try to figure out how they move. She was primarily concerned with how they move their thoracic legs, but the way they move their thoracic legs is intimately related to the musculature that's involved with the spring. Basically, these guys have elongate, rope-like, twisted muscles that run the length of the abdomen. And when these muscles contract, it results in a tremendous increase in the pressure, the fluid pressure, the hemolymph pressure inside the body of the animal. Actually causes these thoracic legs to pop out of their joints. But when that pressure, is released through the release of the furcula from the retinaculum. And so the contraction of the muscles here puts pressure on the furcula, which is then released by the retinaculum, causing the spring to abruptly, explosively move downward and pushing the animal off the substrate, and in this case, pushing it up off to the upper left of this screen. Now, when they jump, they are then totally out of control. Who knows how they're going to land, but they can jump amazing distances, so much so that if you're trying to collect a springtail by chasing one with a let's say a brush wetted with alcohol, they seem to disappear when they jump. They just are able to spring so far away that you totally lose track of them. So it's a very effective escape mechanism. Let's move on to some real photos, I think, of collembola. Yeah, here we go. And so this is a very familiar sort of body type that you find in collembola. And in this family, we see an expansion of the pronotum up over the head. This is not the head. The head is being held down here, the legs here. And this is a fairly prosaic collembola. You see some metallic reflections. Many of these animals have scales covering their body. When you throw these into alcohol, all of this is lost and they just look like pale gray, almost translucent creatures. But when they're alive, they really are amazingly beautiful things. Here's another one. You can clearly see foreleg, middle leg, hind leg. The prothorax is small and reduced here. This is the head, of course. This is the mesothorax or mesonotum, metanotum, and then the abdominal segments. The first, that's where the cauliform is going to be. Second, third, that's where the retinaculum is going to be. Fourth, that's where the furcula is going to be. And then fifth and sixth, toward the end of the animal. Here we get some, and we can point out a couple of things here. These are the segmented antennae. These insects, hexapods, also the Protura, have very simple tarsi, single segmented tarsi, with a single apical claw at the tip. Now we're starting to get into a little color along with the exuberant raised hairs on the body. This is a close-up of what passes for a compound eye here. So we can see 1, 2, 3, 4, 5, 6 ommatidia on this in this cluster. So not the tightly packed ommatidia that are typical of winged insects, but at least the Protura have no eyes. In the next group, we're going to talk about the Diplura also have no eyes. And so at least here we do have the capability of well, I'm not going to jump to the conclusion that simply because a creature does not have eyes that it is not capable of detecting light, but at least here we have structures that are capable of going some distance toward developing eyes that can form an image on them, with them. And then I just had to include some of the really bizarre and beautiful examples of collembola. Again, simple tarsi, and in these groups I've showed you so far, they all have clearly segmented abdomens, maximum of 6 segments. This guy, I'm not sure if this is Podura aquatica. I don't believe it is, but this is pretty much what one of those aquatic ones will also look like. Kind of a vaguely segmented purplish body, nice antennae. Many of them, as I intimated earlier, have actually lost that characteristic furcula, that characteristic spring that one would think would define the suborder. Of course, we believe that the spring is plesiomorphic for the entire order. Recall that in the Rhiniella, the fossil from the Devonian period has a furcula, and so presumably that is an extremely ancient, probably plesiomorphic organ for the order as a whole, or if you want to think of it, it's apomorphic, defining the members of the order. I'm not sure what to think about this thing. It's beautiful in color. It's obviously extremely tiny with little, what would you call them? Setose tubercles arising all over the body. Whether or not these have any function, of course, is totally unknown. But the wild colors that you can find in the collembola are widespread. Again, things that are often lost in museum preserved specimens. But when you see living animals, they really jump out at you. I've always said that the most amazing things are the smallest, and so as you focus in and look closer and closer, you find the most amazing and beautiful creatures. So now I'm going to, the collembola are basically divided into 2 large groups. There's some uncertainty as to the relative monophyly. Probably those I've showed you so far that have the well-segmented abdomens are not a monophyletic group, but that is the plesiomorphic condition. Here we have a family called the Sminthoridae, and this is one in which those abdominal segments have all fused together to form a globose abdomen. It's got this little extra piece back here. Head, the thorax is cramped up in here and fused with the abdomen. These are extremely common, even here in Ohio. These are a very easy family to find. And This may be a monophyletic group. It's probably paraphyletic because the last group of Collembola I'm going to show you probably evolved from this group. But the Sminthuridae is an extremely common family that you're certain to run into. This is the lucerne pest. I'm sorry, the alfalfa pest that I talked about. Lucerne is another name for alfalfa. Belongs to the family Sminthuridae. But let's show you some of the diversity in shapes and colors of these amazing little creatures. Different colors. Sometimes the antennae can be quite elongate. Purple structure, purple color of the body. Again, you can see the loss of segmentation in the abdomen. Mottled colors. Yellows, reds, purples, blues, blacks, everything. I don't know about you, whenever I see a face of a Sminthurid, for some reason it looks to me like a rabbit. But here we see the antennae. Just as in winged insects, though, some of these true segments can also be subdivided into false segments, or what are called antennomeres or flagellomeres. So they can look like they have more true segments than they really have. And this is that final family, the Neellids. Just a handful of species, I think maybe a couple of dozen around the world, that are found in caves. It looks clear to me that this is just a very highly modified Sminthurid, and that's probably what's going on. So Neellids and Sminthurids together probably form a monophyletic group, and then you have the rest of the families of Collembola to deal with. There are lots of families. There's lots of disagreement among the taxonomists on how best to classify them, and so some of these, especially in poorly preserved specimens, can be very difficult to identify to family, and so that's why I didn't push that envelope in terms of the lab and being able to site ID them. The Sminthurids, though, are clearly unmistakable. There's nothing else that looks like them, and so that's a family that you can easily recognize. So let's continue on with the next order of Entonathus insects. Now, the Protura and the Collembola are each other's closest relatives, so now the Diplura are outside of that. Characteristics of the Diplura. They have elongate, clearly segmented bodies. Like the Protura, they have no eyes, but unlike the Protura, and the Collembola for that matter, they have well-developed cerci, that is, posterior appendages that are primarily sensory. Well, let me retract that. That are typically sensory in function, but we're going to see in one group of Diplura that they're taking on another function. I want to mention one fossil from the Upper Carboniferous, a fossil named Testijapix. That root japix, I think I mentioned this last time, is the type genus of the family Gepigidae, which is one of the easily recognizable subgroups of Diplura, in which the cerci, instead of being these elongate antenna-like sensory structures, are more like forceps, just like an earwig. Well, not just like an earwig, but similar to what you see in an earwig, grasping organs at the tail end of the animal. So Testijapix has these modified cerci, but interestingly, they also have well-developed eyes. So whether Testijapix indicates that Diplura ancestrally had eyes and subsequently lost them, or, well, that would be one interpretation of Testijapix. Problem with that interpretation is that reputedly, I have not seen the fossils myself, but I have read some articles about them, and reputedly the fossil is in very poor condition. That is, the definitive structures you would like to see are very difficult to see in the compression fossil, upper Carboniferous. This is going to be a compression fossil. And so it's even up for debate of whether the paleontologist who originally described it correctly, accurately interpreted what she was seeing. This paleontologist was Jarmila Kukalova-Peck from the University of Ottawa. So it's a question whether this is a dipluran or not. And so it kind of begs the question then, even if it does have compound eyes, if that's relevant to what the plesiomorphic condition would have been for the order Diplura. There are two common families or groups of families in these, and the ones you're typically going to run across, and these are fairly common right here in central Ohio. I'm sure that if we were able to get outside and do a little bit of collecting, all it would take is turning over a few rocks in a forested region, and we would find Diplura crawling around under there. The two groups are the Campodiidae and the Gepigidae, and these most obviously differ by the structure of the cerci at the back end of the animal. In the Campodiids, these are elongate, antenna-like sensory structures, and in the Gepigids, they're the forceps-like clasping organs at the back end. The two groups also differ in their basic biology. Campodiids are herbivores or detritivores, whereas Gepigids are active predators. Perhaps somewhere along the line you have heard the term Campodiiform larva used to describe one of the larval types found in insects. And simply what that means is that the larvae look a lot like a Campodiid. That is, they have elongate abdomen, well-developed walking legs, elongate antennae, and such. This is why you would call these a Campodiiform larva. Larva, of course, is going to be in the holometabolous insects, and so it actually has nothing to do phylogenetically with the Campodiidae. It's just that the larvae look something like one of these diplurans. You can find Diplura, as I mentioned, in the soil, underneath rocks, in rotting wood, in the leaf litter. They're not at all uncommon. So I'm sure you would be able to collect some if you were able to get out into the woods these days. So here's a typical Campodiid. These are the elongate cerci. You can see they look pretty much exactly the same structure as you see on the front end of the animal with the antennae. No eyes. As we saw earlier in that SCM, the mouth parts, the maxillae and the mandibles, enclosed within the lateral folds of the head capsule. Three well-developed pairs of thoracic walking legs. And in the next few pictures, I'm going to be able to focus a little bit more on what's going on here in the abdomen. But this is typically what you would see. And in terms of size, this would be, 1, maybe 2 millimeters long. So they're small. They're small creatures. Here we focus in a little bit more, and now you can see on each of these abdominal segments there is what's called a stylus. Now maybe you're familiar with the term stylus from in the context of as a pen, but a stylus is a modified unsegmented appendage. In this case, these are abdominal styli, so these are occurring on the abdominal segments. What, if any, function they serve is unknown. Perhaps they help to support the body of the animal. It's unclear what they do. Here's a even closer look at these. You can see they're musculated at the base. That this one is promoted, moved forward in relation to the body, whereas this one is remoted. So you can see that they're moving, perhaps they're supporting, perhaps they're contributing to locomotion a bit, especially if you think about the animal wriggling through the crevices in the soil. You can perhaps imagine that these things help with locomotion to some extent. Here's one with I blocked the antenna here, but you can see the extremely long cerci, these modified appendages. what's the word I'm searching for, canonically associated with the 11th abdominal segment. That is the terminal abdominal segment in all hexapods except Protora. So those are the Campodeids. Oh dear, let me see if I can move my picture. I can't move my picture here. But you'll be able to see it on the PDFs that I'm going to post of this slide. This is a Gepigid in which the cerci at the back end here, it's a shame that my ugly face is in the way, are modified into forceps-like structures that can pinch and grasp. These are predatory orcas. They're not nearly as common, at least in my experience, as Campodeids, but they are found here in central Ohio. Okay, so now we're going to move out of the Entognathus hexapods and into the Microcoryphea, or the jumping bristletails. As I said earlier, this group of insects in the past was often grouped together with the Thysanura, which is the next we're going to talk about. But there are a number of differences between them. One of the most obvious is the jumping ability. That Microcoryphea typically have this hunchbacked appearance with the same sort of strong longitudinal muscles that you find in Columella that enable them to do this jumping. They have well-developed compound eyes, just as do many of the Thysanorans, but in this case, the Microcoryphea, the eyes are so large that they extend dorsally and meet along the dorsal midline of the head. They have abdominal styli, just as you saw in the Diplora just a moment ago, but they also have styli on the coxae of the metathoracic legs. So on the hind legs, on the coxa, there is a stylus there. This is consistent with some of the ideas about the fundamental structure of an arthropod leg. That is, that it is multiramus, or perhaps even biramus. Remember that Manton's hypothesis was that the hexapods and the myriapods and Onychophora for that matter, are characterized by having primitively uniramus legs, unbranched legs. Well, the presence of styli on the hind coxae of the Microcoryphea adds an element of doubt to that. It's evidence that they are not, that the uniramus legs are not the ancestral condition, that they may have had what are called excites on these leg segments. And it's from excites that one theory of the origin of wings says they came from, that they are a combination of the thoracic wall and perhaps the notum, but the excites, the styli in this case, are the where wings develop from. Both Microcoryphea and Thysanura have well-developed elongate cerci, along with a central filament. I'm not going to reprise the Monura that we talked about in the context of paleoentomology, but that was an Upper Carboniferous group of insects that had only the single median caudal filament, caudal meaning tail, and the cerci were still in the form of those small leg-like structures. In Microcoryphea, we find something that is not present in the three orders we've talked about so far, and that is what's called an appendicular ovipositor. In other words, modified appendages on typically the 7th and 8th abdominal segments that are used to assist in oviposition, the placement of the egg. In both these groups, though, we still retain the indirect sperm transfer that we found in the Entognathus hexapods. There aren't a whole lot of species of Microcoryphea, only about 500 species worldwide, and they're usually pretty secretive and nocturnal. You can collect jumping bristletails here in central Ohio, down in the Hocking Hills. Typically you put out traps and they'll collect themselves at night. But so they're not something you're going to really see, and because they can jump, they're very difficult to collect by hand. You really need to set traps in order to capture them. In terms of what they are doing in the environment, they're typically grazers or scavengers scraping up food particles off of substrates. One of the most well-known species, Petrobius maritimus, lives in the intertidal zone and feeds upon algae that grow on rocks, scraping the algae off the rocks and grinding them between their mandibles. I guess I jumped right over that mandibular section, right? We're still in the position, as I described last time, of having monocondylic mandibles. But I've got some images to show you that'll probably make their structure and function a little bit clearer. So these are drawings taken from Sidney Manton's groundbreaking work that was published in 1964 on mandibular mechanisms and the evolution of arthropods. And this is the head of Petrobius maritimus. All the pictures are of Petrobius. This is what it looks like from the outside. And we see this is the base of the antenna here, the large compound eyes here, the tergum of the first thoracic segment, and down here are the mouth parts. Let's see, what do we have? We have the labium back here, and this is the labial palpus. This is mostly in the unstippled portion here, the maxilla with the maxillary palpus arising here. And as you'll see when we see whole photographs of the entire body, the maxillary palpi in microcoryphea are actually quite large. Underneath here is the mandible. It's not clear in this view, but here Manton has dissected away some of the pieces. And this dotted line here that begins here and reaches up in this area, following the course of my cursor there, that is the mandible. And this dot right there where my cursor is, that little dot is the dorsal articulation of the mandible. So that's where the mandible is attached to the head capsule. And then it has muscles indicated by these hatched lines here that insert on the posterior and on the anterior surface. And when these contract, the mandible rotates back and forth around that dorsal articulation. Here what Manton has done, and these are Manton's illustrations. Manton has dissected out, figuratively speaking, the mandibles. This is the dorsal articulation here, where it's attached to the head capsule. See, they're long, pointed at the tip, and they can be protruded to some extent, just as we saw in the Entognathus hexapods. But along their mesal surface, so along the midline, there is an expanded lobe here. And so the movement is this basic promotor, what she called rolling motion of the mandible. So in this view, we'll have muscles attached along this edge and this edge. In this picture, they're very close together, but it causes the mandibles to rotate along an axis that is more or less vertical here, going from the tip to this point of articulation. And in doing so, it causes these mesal lobes to rub together mesally. So it's not actually biting transversely, but it's rubbing those mesal lobes together in a grinding motion. And so basically, this animal would scrape up algae off the rocks with some saliva, suck it in between the nathal lobes here, and then grind it before ingesting it into its body. And so that's the plesiomorphic way in which the mandibles function in microcoryphea to start with, and then in the rest of the hexapods. Except in the rest of the hexapods, the insects that we're going to get into, we're going to add a second articulation posterior to this dorsal articulation here. So here is a jumping bristletale. As I mentioned, it's got this kind of humpbacked appearance there, which is very typical of them. Covered with scales, typical. What else can we see here? These are the maxillary palpi, so you can see they're quite elongated and very similar to legs in structure, basically. The styli here, you can see the styli on the hind coxa and actually on the mid coxa as well. So we have to account for that in our theories of what the fundamental structure of a hexapod limb or an arthropod limb looks like. Large compound eyes. Let's take a look at a different one. what can we see here? Here I've chosen a picture that emphasizes the presence of the abdominal styly. Here's one here. Here's one here. Can we see any others? Not too clearly. Maybe one right there. Here's the cerci and the elongate caudal filament coming out the middle. Again, that typical humpbacked appearance, scaly body. So modified, flattened setae to form scales on the body. Again, coxal stylus on the thoracic leg, and the abdominal styli. We can see 1, 2, 3 of them here. I believe this is the ovipositor of this creature that are also modified appendages of the abdomen. In the protora, diplora, and columbula, there is no appendicular ovipositor. Basically, the eggs are deposited directly out of a gonopore. There are no appendages to assist in placement of the egg within the substrate. And that leads us to the final order in the apterygote insects, the Thysanura, or what's also known as the Zygentoma. These are characterized by true transverse biting. That is, a second point of articulation has been made between the mandible and the head, and now the mandible rotates about along an axis that connects those 2 points of articulation. And so we had one dorsal, now we're adding a second one, and now the mandible is going to rotate about that. Thysanura are actually very common, and some of them are household pets. You're probably familiar with firebrats or silverfish that are, feed upon a variety of materials. Silverfish are notorious for feeding upon starches, so your bookbinding or wallpaper paste or stuff like that is a glorious buffet for a silverfish. But there are also some that are herbivorous. There are even some Thysanura that live as inquilines within ant nests, and what exactly they're doing there is still up a question that needs to be answered. There aren't a whole lot of species of Thysanura worldwide. There's only like 310 species, the vast majority of which occur in the family Lepismatidae, which is the same family that the silverfish, Thysanura, and the firebrat, what is the name of firebrat? I don't remember off the top of my head where they occur. There is one interesting family that occurs in the United States, the family Lepidotrichidae, which is known worldwide from only a single species, which is found in California. Tricholepidion is the name of the genus. Don't worry, you don't have to remember that, but Lepidotrichids are interesting in that they retain a number of what appear to be plesiomorphic characters for Thysanura as a whole, whereas something like a silverfish or a firebrat has a reduced number of tarsal segments and no ocelli. Lepidotrichids retain ocelli, the typical 3 ocelli of an insect on the head, in addition to the compound eyes, and also a 5-segmented tarsus. Well, only 2 of those are true segments in the sense that I earlier described, but the subsegmentation so that it appears to be 5-segmented in the tarsi. And also I should note that both Microcoryphea and Thysanura have double claws, ungues, at the apex of the leg on the pretarsal segment. So Lepidotrichids retain a number of characters that are plesiomorphic, and it was thought at one time that they may be important in understanding the phylogeny of insects as a whole. While they may have some potential, and for example, E.L. Smith used them extensively as a model system for understanding the evolution of the insect ovipositor. These days, the molecular data have largely overwhelmed the struggle to recognize homologies and to find characters in morphological characters as a key for morphological structures as a key for understanding phylogeny. And so the critical nature of Lepidotrichids is a little bit passé, as it were, now. And also, if you look closely at the Misoff et al. article, the One Kite project, you'll find that their data indicate that Thysanora are indeed a monophyletic group, so that the Lepidopterygotes form a monophyletic group together with the more typical lepismatids, and that actually they don't have anything. The Lepidopterygotes themselves are not the sister group to the winged insects, but the Thysanora as a whole are their sister group. So quickly taking a look at some images. This is a lepismatid. The front end is up here. We can see the abdominal styli. They're very characteristic. Circe, appendicular ovipositor here. The caudal filament is blocked in this picture, but typically these guys are quite flattened. And in fact, the cocci, instead of protruding ventrally from the thorax, the cocci of the thoracic legs that is, are shifted to lie flat. And so not only the body, but everything about this creature is modified to fit into very narrow spaces. I just wanted to show you one to indicate that these can be a little bit more colorful and interesting than a typical silverfish or firebrat. What can we see here? The elongate fringes on the front of the head, antennae, thoracic legs, and then it's a little bit out of focus in this image, the caudal filament, the median tail filament, and one circus on this side and the other circus off at an angle on the other side. So to summarize, let's see. For the apterygote insects, again, apterygote insects do not form a monophyletic group. So this is simply a grouping of convenience to take some things that have a lot of plesiomorphic characters and relatively few species, and to be able to talk about them here as a unit. There are the Entonathus species, and again, either Entonathi evolved separately in the Diplura and the Columbella plus Protura, or Entonathi would be a plesiomorphic character that would have characterized the common ancestor of all hexapods. The consensus of opinion, I think, is that the Entonathi of Diplura and that which we see in Columbella and Diplura and Protura evolved independently. And so Entonatha is also another informal group of convenience that should not be interpreted as indicating anything of evolutionary significance. The Protura are characterized by lots of things, but the most obvious excuse me, is that they have no eyes, no antennae, and you can go back to the part of the video in which I discussed Protura to get some other characters for them. Columbella, the really unique thing about the Columbella, when it's present, is that furcula, that structure off the fourth abdominal segment that enables these creatures to spring, to jump as as an escape mechanism. Columbella have antennae, but they are segmented antennae, meaning that they have intrinsic musculature within what appear to be the antennal segments. Diplura also have segmented antennae, but interestingly, they have no eyes, but they also have well-developed cerci, which are not present in the other two orders. So we kind of have this set of characters, and they come in combinations. No eyes in Protura and Diplura. No segmented antennae in Columbella and Diplura. Cerci, excuse me, only in Diplura. So they come in different combinations, and as I said in the last video, every possible way of grouping these things together to understand their relationships has been suggested, and there is evidence for it, but the weight of the molecular evidence at the moment comes down on the side that the Protura and the Columbella are each other's closest relatives, and Diplura are the closest relative to all the rest of the insects. The microcoryphia, or jumping bristletails, have still the basic plesiomorphic monocondyllic mandibles. Everybody else we're going to talk about from here on out has dicondylic mandibles. And the first group we talked about there was the Thysanora. And the Thysanora form the sister group to the vast array of insects we know as the pterygotes, or the winged insects. So we're going to call it a day here. Next time we'll jump into the modern Paleopterous orders of insects, the Ephemeroptera and the Odonata. Until then, let's hope the weather turns better, no snow, and maybe we'll meet outdoors in my backyard.